Hello, everyone. I'm m.stemkovski. I'm a PhD student at Utah State University, and today I'm excited to talk to you about bee phenology in the Rocky Mountains. And let's get right to it. So this work was done at the Rocky Mountain Biological Lab in Gothic, which is right up the road from Crested Butte. Some of you may have been there. We love working there because it puts us in these beautiful meadows of long-lived perennial flowering species. But that's not the only reason. The growing season there is very short. There's a very narrow window between when the snow melts in the spring and when it comes back in the fall. And that really compresses things. It compresses the flower community and the bee community down to a digestible package that we can study. So before I go any further, I want to acknowledge the many people who have contributed to this project. There's been lots of people collecting data as well as making sense of it, and as well as our various funding organizations and institutions that have made this all possible. So a brief outline of what I'll be talking about. I'm going to introduce you to phenology, why we should care about it. I'm going to talk about the possible drivers that have been hypothesized in the literature of the drivers of bee phenology. I'm going to talk to you about how we went about tracking bee phenology and then some of the statistical methods to get at what's driving that. I'll talk to you about the results and the takeaways and come back to the question of what can we expect with climate change. So what is phenology and why do we care about it? Well, phenology is simply the timing of life history events, the yearly timing. We're all intimately familiar with this actually. So in the spring, leaves come out, they turn green and they grow and they mature. That is Phenophase, and there's a time associated with that, but it varies year to year, sometimes depending on what species you're looking at, depending on the climatic conditions, depending on all number of things. And what we're seeing right now, I'm recording this in the fall, is the phenophase of leaf senescence, when those leaves are turning brown and falling off the tree. That also has the timing associated with it, which is predicted by a whole nother suite of things. But insects and bees specifically also have phenology, have yearly phenology. You can think of it as the timing of when an egg is laid, when that egg develops into a larva and then into pupa and then finally into an adult. Most of you probably have not seen larvae and eggs and pupa of wild bees, and neither have I. They're very cryptic. And so in this talk, we're going to be focusing on the adult stage. That's what's actually out there interacting with flowers. So let's get a historical look at what is phenology supposed to look like from the bees and the flowers perspective. Well, from a flower's perspective, a flower opens, it offers up its nectar and pollen resources in exchange for pollination services from bees. And if all goes well, flower blooms in the same time as when bees are there to forage on it, and that results in a healthy seed set. From a bee's perspective, a bee wants to time its foraging to the time when its preferred floral resources are open, and that allows it to have high fecundity. But if bees and flowers change their phenology in response to something like climate change at different rates, there can be a phenological mismatch where a flower opens, but its pollinators are nowhere to be seen. They have not emerged yet. They have not started foraging. And from the bee's perspective, a bee emerged, but it's too late. There's no flowers and or not enough flowers, and that results in smaller fecundity. So what are the possible drivers? What predicts bee phenology? There have been studies, usually on small subsets of communities, just one or two species, and looking at certain variables or traits in isolation. The goal here is to look at everything in unison, figure out what is important, put it all in the same scale. But let's run through some of the things that we have found from the literature to be important. First and foremost, and probably most obviously, elevation in this mountainous area is important. The growing season starts sooner at lower elevations and it lasts longer, typically at lower elevations than at high elevations. The degree of solar incidence is also important, and that's just how directly a slope is facing uh, the sun. So if you have a site on a southern steep slope, then that's getting a lot of direct sunlight. Whereas if you have a site or a bee nest on the north side of a slope, it's getting a lot less heat and light. There's a number of climate variables that are probably important to bee phenology in this mountainous, snow-dominated system. Snowmelt, the timing of snowmelt is probably very important. Rainfall has been shown to be important, as well as just temperature directly. And then there's also a number of species traits that there's good reason to think might be important for phenology. First is size. Bees come in all kinds of shapes and sizes, and size in part determines their thermal limits, their thermal thresholds. So there's reason to think that at different times of the season, different sizes of bees might be able to forage. Bees also vary in their nesting location, and this can be boiled down to below and above ground nesters. Those that nest below ground are in this snow-dominated area spend most of their life covered by a blanket of snow, whereas those above ground are exposed to the variations of air temperature. And lastly, the life stage in which bees overwinter is probably very important too. Some bee species in the year that their eggs are laid go ahead and develop through those stages of larva, pupa, and then get it all the way to fully formed adults. But they just don't emerge from their nests until the following year. They just hang out, so to speak. Other bee species, however, overwinter as pre-pupa. So when the next spring rolls around, they still have some developing left to do before they can start emerging and foraging. So how do we estimate? We know what might drive this phenology. How do we actually gather data on it? How do we estimate using the data that we gather what determines phenology? So first, in 2009, Becky Irwin and her team started a project to monitor bee phenology at the RMBL. And this is paired with a long-term flower phenology monitoring project that was started in the 70s, which I will come back to. The basics of the method, of the sampling method, are we put out these brightly colored bowls. They're about this big, I guess. And they're filled with soapy water, and they're colored white, fluorescent yellow, and blue, which are attractive colors to bees. They mistake them for flowers. And then because they're filled with soapy water, they get caught in these bowls, and then we can take them back to the lab and identify them to species, something that we are not able to do out in the field. There's lots of species and they look very similar. So we've been doing this every year since 2009. The data I'll be showing you is up to 2017. We put out these pan traps every 2 weeks at 16 sites along a roughly 1,000-meter elevation transect. And after all is said and done, we've recorded about 150 species in just this one area of Colorado. So after all of that, we can aggregate this data, and this is roughly what it looks like. So this is just for one species at one site in 1 year. We have day of year on the x-axis, so that's just days since January 1st, and then abundance on the y-axis. And you can see that, well, there's not a whole lot of data points, right? 2 weeks is not all that often when you have such a short, narrow growing season. And so we need some kind of method to be able to intelligently estimate when bees started foraging, when they peaked, and when they stopped. So I'll save you the details of the statistical method. I'll point you to where you can read more about that if you're interested. But basically, we fit a model based on generalized additive models. We calculate the uncertainty around that. And then we calculate the emergence, peak, and senescence time based on that prediction, and then uncertainty estimates around those predictions. So keep this in mind. There's three phenophases that we talk about: emergence, which is when bees started foraging; peak, when they peaked in her foraging activity; and then senescence is when they stopped foraging in the season. And so we do this for every combination of species by site by year, and we get our data. We get our estimates. And so then what do we do with it? Well, step 2 of the statistical analysis is to take all of those potential drivers that I already mentioned, those climatic, topographic, and species trait drivers, and plug them into 3 separate statistical models to predict emergence, peak, and senescence. I won't go through this because I actually already have. The only thing to notice here is that we're treating species and site as random effects to account for pseudo-replication at the species and site level. We weight this regression by the confidence intervals to propagate uncertainty. Again, you can read more about that another, I'll send you the paper. And we do model averaging using mu-min, which I will also not delve into. So what do we find? What determines bee phenology? Remember that we want to compare all of those variables against one another, and so we need to standardize them, to put them on the same scale so we can answer questions. How much more important is elevation than body size? You know, from individual studies that look at just one species or one variable, you can say that, yes, this is statistically important, but you can't say this is more important or this much more important or effective than the other variable. So this is the main take-home figure, which I will break up into chunks, but to prepare you for interpreting it, on the y-axis we have standardized effect size. That's just the common scale across all these effects. And on the x-axis, we have those different variables that I've already introduced you to. If you see effects above 0, greater than 0, that means that that effect causes, or that variable causes those phenophases to be delayed. If you see values below 0, then that means that those phenophases are getting earlier in the year. So let's look at climate first. So you can see, first and foremost, snowmelt plays a big role. The later that the snow melts, the later all of these phenophases occur. That's what you might expect, but then if you look more closely, you can see that emergence and peak timing are more affected than senescence timing. That makes sense when you think about it, because once the snow melts, the first thing that happens is the bees emerge, you know, and there's a few months, it takes a few months for them to then senesce. And you can see that similar kind of pattern for these other climatic variables, where the further you go along from emergence to peak to senescence, those absolute effects of these climate variables get smaller. It looks like emergence timing is most determined by climate. Let's look then at topography. Not surprisingly, elevation plays a large role. So the higher up you are, the later bees emerge, and also the earlier they senesce. So in other words, the bee foraging season is narrower at higher elevations than at lower ones. And then solar incidence plays some role, but not as much. We can move on to traits. And traits are important. Body mass, not so much, so I won't spend time talking about that. But then nest location and overwintering stage are influential. And let me just clarify how to read this. So these are contrasts between two different levels. Nest location, if you remember, can be below or above ground. And so here what you're seeing is the bee species that nest below ground compared to those that nest above ground. In other words, bees that nest below ground, they emerge later than those that nest above ground. And you can read overwintering stage the same way. Those that overwinter as pupa, they senesce later than those that overwinter as adults. So, we've calculated these standardized effect sizes, but some of you are probably asking, well, how well did you actually predict this? Did you explain like 5% of the variation or how much? Well, we actually explained quite a bit of the variation because this is a mixed model. We have both marginal and conditional R squared values, but they're both pretty high, at least for an ecological study. And there's also not a clear trend for us being able to predict emergence, peak, or senescence better than the other. The model does pretty well for all three phenophases. But then the story becomes a bit more nuanced. When you try to predict these phenophases using just the climate variables, excluding everything else, you see that our ability to predict these phenophases decreases as you go across the phenophases. So in other words, climate explains a lot more variation in emergence than it does in peak or senescence. And then the opposite is true for traits. It looks like traits are the main thing, species traits are the main thing that determines when species senesce in the fall. And that's the opposite from when they emerge. Looks like that is much more climate driven. So what about trait by environment interactions? This is the kind of interesting ecological bit. Are species with certain traits more responsive to variation in climate? And it looks like yes, though not as much as you might expect. So what I'm showing here is nest location interacting with snowmelt timing, and it looks like, at least for the emergence phenophase, those species that nest above ground are more responsive than those that nest below ground. There's that difference in slope. This might seem unintuitive because those that nest below ground, those are the bees, those are the species that are covered by a blanket of snow, so shouldn't they be more responsive? But it might make a bit more sense when you think about what's driving snowmelt timing to begin with. Spring temperature is a big driver of how fast that snow melts, and those species that are nesting above ground are the ones that are experiencing those variations in air temperature. So this pattern might actually make sense when you think about it that way. And then secondly, we can look at overwintering stage interacting with snowmelt, and there's not, we did not find significant interactions, which is somewhat surprising, and definitely more work needs to be done here. I hope this is a provocative result for some of you. But at least this study suggests that bees that winter as adults and those that winter as pre-pupa, they are equally responsive to snowmelt, at least. So back to the central question, is there reason to expect a phenological mismatch between bees and flowers on the community level? Well, we can look back at that flower phenology study that I mentioned that's been going since the 70s and compare our findings to that study. So to summarize, in 2014, a study found that the date of first flowering advanced by 0.89 days per day of advance of snowmelt timing. So it was tracking, but not perfectly. And then the two flowering peaks, there's two in this part of the Rocky Mountains, were 0.74 and 0.53 days, respectively, in comparison to 1 day of snowmelt time in advance. Now, when you compare that to those same numbers from our study, we found that bees advanced their emergence by 0.49 days per day of snowmelt advance, which is substantially less responsive. Peak is also substantially less responsive. And when we when we break it up a little bit, when we look at different species and make some species-specific estimates of their rate of response to snowmelt, this is a busy figure, but all you need to know is that if a point is on 0, that means that that phenophase is not responsive at all to snowmelt timing. If a phenophase is at 1, that means that it's perfectly tracking, and if it's in between, then it's somewhere in between. So most of them fall in between. So they're tracking snowmelt, but not perfectly. And then when you plot these flowering averages onto this figure, so this is the first flowering day, you can see that almost all of these shifts in bee phenophases fall to the right of that. So they're not keeping pace. Similar story for peak phenology, though it's maybe not as dire. This leads us to think that yes, we might expect a mismatch between bee foraging and flower blooming. It seems that bees are not able to be as responsive, at least to snowmelt timing in this area. So some key takeaways. First, everything's important. We helped confirm that all those variables that we thought were important, but some are more important than others. In particular, climate was very important in predicting early phenophases. And then that species traits were more important for predicting later phenophase, especially the stage in which bees overwinter. And this study leads us to suspect that there might be a community-wide mismatch between bees and the flowers that they try to forage on, and that some species, based on their traits, are going to be more affected by this than others. So where do we go from here? Well, I used a lot of words like may and suggest here. To be more sure about how bees and flowers are going to be impacted by mismatches, we need to actually predict the fitness effects of mismatches. That's one of the next steps. And then another one that I'm currently working on is determining the shape of bee phenological curves. The statistical method, the generalized additive models, are specifically designed to be sort of agnostic when it comes to curve shape. They're supposed to fit anything pretty well. But part of understanding how mismatches are actually going to affect fitness is being able to understand what does a typical phenological distribution of bees and flowers looks like. If I've piqued your interest, hopefully I have, and you want to read more about this, more about statistical details or just details about the findings in general, I'll point you to our recently published paper on these findings in Ecology Letters. You can find a link to this in the description of this video. And if you have questions or comments, I would love to talk with you. So feel free to email me at this email, and you can also find this email in the description. And thank you for listening.